DNA, Structure and Replication by Isabel and Claire. Once upon a time, there was adenine, thymine, and cytosine, and, of course, guanine. Adenine and guanine are purines, and thymine and cytosine are pyrimines. Phosphate and sugar are also present. They were the best of friends, and they always stuck together, literally. But adenine always paired up with thymine, and cytosine always paired up with guanine. Phosphate was best buds with sugar, and they're known as complementary pairs. Thus the order goes: in a 5' to 3' direction, the leading strand with the phosphate, in an anti-parallel pattern. They create a double helix formation, caused by nucleosomes, which are DNA with 8 histone proteins held together by another histone protein. Zooming in, we have a polydiester linkage between the phosphate and the sugar, and covalent bonds between the base and the sugar. Bases have an entirely different reaction, though. They are connected by hydrogen bonds. Adenine and thymine all have double bonds, and guanine and cytosine have triple bonds. They also like to replicate by having helicase men split their bonds headed towards the replication fork. Replication of DNA always begins at special initiation points, or origins of replication. In eukaryotes, because they're complex, there are multiple initiation sites, whereas prokaryotes, being blobs, only have single initiation sites. Then, DNA polymerase III adds nucleotides from a 5' to 3' direction on the leading strand. However, before we start this, we need an RNA primer, headed down by primates. DNA polymerase III adds to this and completes the strand, while something else happens to the lagging strand. RNA primase comes and adds a short length of RNA, RNA being a primer. Afterwards, zooming in, DNA polymerase III starts replication in front of the RNA. Notice the Okazaki fragments are the DNA between the RNA and DNA polymerase III. 5' to 3' direction is the key. The DNA polymerase III wants to go back to the replication fork and does that by attaching to another RNA, creating another Okazaki fragment. DNA polymerase I then comes in and removes the RNA primer and replaces it with fresh DNA. However, there's still a gap between the two nucleotides. So, zooming in again, DNA ligase seals up the gap with a sugar and phosphate bond. Voila! We're done with the semi-conservative replication! Wait, what's that? Oh, sorry, my bad, I didn't explain. Well, it's our double helixes, but they consist now of one parent strand and a daughter strand. Oh, the old and the new. Yeah, technically what I just said. Now for some fun. Helicase man! Meow.